Hi everyone, we're iGEM Aachen and we're super excited to share our project OncoBiotica with you today. We hope you enjoy. For decades, cancer has loomed as a global health crisis, creating immense challenges for healthcare systems worldwide. In 2020 alone, cancer claimed over 10 million lives, making it the leading cause of premature death. In response, the United Nations has set an ambitious goal to reduce the amount of premature death from non-communicable diseases by 1/3 by 2030. Continuous innovation and research are essential in this fight against cancer. Today's advancements are paving the way for more precise and effective treatments. The future lies in precision medicine, where treatments are tailored to each patient's specific profile. OncoBiotica stands at the forefront of this shift by offering a groundbreaking approach for precision medicine. With emerging research on tumor-associated microbes and the growing need for novel therapies, OncoBiotica redefines the current landscape by focusing on an untapped target in tumor therapy, the microbiota within tumors. Our approach is different from conventional targeted therapies, which focus on molecular biomarkers of the cancer cells. Instead, we focus on the microorganisms within the tumor's microenvironment. This approach leverages the unique differences between microorganisms and cancer cells. Our two-phase mechanism begins with the introduction of a fusion protein, which selectively binds to the bacteria in the tumor. In the second phase, a prodrug is administered, and it's converted into a chemotherapeutic agent right at the site of the tumor, effectively eradicating surrounding tumor cells. We've chosen colorectal cancer and its association with Fusobacterium nucleatum as our model system. Colorectal cancer is the second deadliest cancer globally, and while incidences have stabilized in higher income countries, cases are rising among the younger population and in many low and middle income regions. Fusobacterium nucleatum is an anaerobic gram-negative bacterium found in the tumors of these colorectal cancer patients, and therefore we believe it's an ideal target for testing our therapy.
Early on, we focused on gathering insights from a diverse pool of experts from both industry and academia. This approach provided us with valuable perspectives on the feasibility, marketability, applicability, and overall project development. The exhibit interviews offered crucial input for our laboratory work, hardware design, and modeling efforts, which allowed us to refine our project at various stages. By engaging with experts from different fields, we ensured that our project, that we covered every aspect of our project through a deep dive, considering multiple angles. This early and consistent expert feedback was key to shaping our product direction, ensuring that it was built on a solid foundation. Additionally, we integrated human practices from the start, allowing expert insights to directly influence our project directions.
To create the fusion protein, we designed a biological part, encoding a binding domain already described by a previous iGEM team, a linker and an enzymatic domain. This second functional domain is the COD-A cytosine deaminase, which is native to E. coli. In its host organism, it converts cytosine to uracil, but it is also capable of converting the non-toxic 5-fluorocytosine into the active chemotherapeutic 5-fluorouracil. Using this prodrug enzyme approach, we showed that our fusion protein is functional. Before starting in the lab, we evaluated the protein design with protein structure prediction. The fusion protein was then produced in E. coli and purified using affinity chromatography. Since the fusion protein is designed to target cancer-associated bacteria, we created a reporter version to evaluate its binding affinity to different strains of Fusobacteria ourselves. To do so, we used flow cytometry. These assays indicated a potential binding interaction. While our initial studies have provided valuable insights, there are still areas for future investigation. One priority is the development for a more robust assay for quantifying the binding affinity to Fusobacterium. To assess the catalytic activity of the fusion protein, we conducted enzymatic assays using high-performance liquid chromatography and nuclear magnetic resonance. For further enhancement of the fusion protein's turnover rate and substrate specificity, we researched literature for mutations that improve enzyme activity. Using computer-assisted recombination, we found stable mutation combinations. Structural modeling of these mutants' interactions with the ligand helped us narrow down a few enzyme variants with potentially optimized performance. Next, we expressed 5 of these variants and analyzed their enzymatic activity using HPLC and NMR. Activity assays identified 2 mutant variants with approximately doubled catalytic activity compared to the wild-type fusion protein. Our novel gamma variant with a second mutation is expected to have a potentially higher relative affinity to our substrate 5-rho-cytosine over cytosine, making it our most promising candidate. This increase in relative affinity is indicated by our dry lab results. Given the modular design of our fusion protein, we can explore different binding domains to potentially enhance its binding affinity to Fusobacterium or target other tumor-associated microbes. By exploring different options, we can optimize the efficacy and safety of the fusion protein. In conclusion, our research has laid a solid foundation for the development of a novel microbiota-directed cancer therapy. The beauty of our design lies within its modularity. With relative ease, the binding and enzymatic domains can be interchanged depending on the target organism or the desired therapeutics. By addressing the remaining challenges and pursuing future research directions, there's hope of translating our findings into a promising therapeutic approach.
As part of our iGEM project on targeted cancer therapy, we developed a hardware solution to advance personalized cancer treatment, a research field growing rapidly worldwide. This approach is particularly important for making cancer treatments more accessible, especially in developing countries, where affordable healthcare solutions are crucial. Our goal is to create a system that is both effective and cost-efficient. At the core of our hardware project is Mike, a device designed to assess the effectiveness of cancer treatments. For Mike, we specifically designed a microfluidic chip which serves as the platform to culture colorectal carcinoma cells. This ties directly into our overall project, where colorectal cancer serves as the model. Mike is a compact system equipped with LEDs and color sensors to measure the color change, which our custom software then uses to determine cell viability in both chip chambers. This allows us to quantify how effective the tested drug is. To ensure accessibility, we considered cost-effective manufacturing methods throughout the whole process. The chip features 2 chambers, one for culturing untreated cells as a reference and another for drug efficacy testing. The chip is inoculated with cells from a pre-culture, and after a 24-hour incubation period, the drug is introduced into the test chamber, while the reference chamber remains untreated. In our proof of concept, we used ethanol. Then, both chip chambers are filled with a risazurin solution, a dye that changes color based on cell viability. And after a short incubation time, the chip is then placed in the Mike measuring unit. With our approach, we could demonstrate a clear correlation between risazurin color change and cell viability. Additionally, a linear relationship between the risazurin signal and cell numbers confirmed Mike's ability to quantify cell viability. Through multiple iterations, we optimized both the chip and the Mike system. We successfully cultured CRC cells in the chips with no difference in growing or adhesion compared to standard cell culture dishes and flasks. Our microfluidic chips and Mike are highly modular, adaptable for various applications, including different cultivations and colorimetric assays. Looking ahead, developing culturable cell lines from patient-derived tumors will be key to expanding our concept for broader use in personalized cancer therapy. The system's cost-effectiveness makes it ideal for resource-limited settings, democratizing access to treatment. With the ability to collect anonymized data, to recognize patterns, it could significantly speed up diagnostics and therapy decisions. Our vision is to make personalized cancer care accessible globally, bridging the gap between advanced research and practical healthcare.
Our dry lab was greatly enhanced by modern AI tools. AI isn't just changing the future of biology, it's revolutionizing it. Tools like AlphaFold have made leaps that were once thought impossible, giving us unprecedented insights into protein structures. But here's the catch. While many biologists are quickly learning how to use these tools, there's been a massive blind spot in teaching how to build them. Imagine the possibilities if more minds were equipped to dive into the very code that drives these breakthroughs. That's where AlphaFold Decoded comes in. We are here to demystify AlphaFold, breaking it down to its core so you can rebuild it yourself, piece by piece. AlphaFold Decoded has 3 main components. First, we've developed a full implementation of AlphaFold in PyTorch, focusing on readability and education. Our implementation condenses the core functionality of AlphaFold into 1,400 lines of code, compared to 5,300 in a selection of key files from the original repository. While other open source versions aim for customizability or efficiency, resulting in a larger code base, ours closely follows the pseudocode from the original paper. This approach provides a direct and clear path from theory to practice. Next, we offer guided learning through Jupyter Notebooks. These notebooks walk you through every step from implementing AlphaFold, providing concrete information on each method and module. We give you skeleton code for functions, but you have to implement all of the logic yourself. Included in the notebooks are automated error checking cells that compare your results with pre-computed outputs. To ease setup challenges, we made our notebooks fully compatible with Google Colab. This means you can work on publicly available machines for free without the hassle of setting up a high-performance environment on your own device. The third part of our project focuses on how we bring this material to the community. We started with offline workshops at our own university, inviting a diverse group of both biology and computer science students to participate. We also presented AlphaFold Decoded at international iGEM meetups, as some of you might remember. The response you gave us was overwhelming and a major driver for us to continue with this project, but it also showed us that we really needed to switch to online material. So we adapted. We switched to hybrid workshops and eventually developed a 9-part video series on YouTube. This allows students to work on the material at their own pace, wherever and whenever suits them. Now, let's talk about the content of the series itself. Building AI tools for structural biology is complex, but we've crafted a clear roadmap. Starting with the basics, we assume only basic Python knowledge and cover key prerequisites like PyTorch, machine learning fundamentals, and attention mechanisms. After that, we dive into AlphaFold-specific content, from feature extraction to the evoformer and the structure module. By the end, you'll have built a fully functional AlphaFold version that's capable of inferring protein structures. Our goal is to make this high level of understanding accessible by providing a concrete pathway that turns a complex topic into an achievable learning experience. This project isn't just about learning. It's about building a platform for the next generation of scientists to actively contribute to the AI revolution in biology.
In addition to advancing technical skills through AlphaFold Decoded, we placed strong emphasis on community outreach and education. Our outreach included 42 interviews, 2 school visits, 4 AlphaFold Decoded workshops, and participation in 4 city fairs, 8 national and international meetups with fellow iGEM teams and external stakeholders, as well as 3 industry and startup fairs. With our participation in the iGEM Startup Program, we actively engaged in industry orientation and business planning. These efforts ensured consistent advancement of our project. Through various initiatives, including workshops and presentations that promoted synthetic biology and STEM, we focused on raising awareness of life sciences and cancer and making science accessible and inspiring to diverse audiences. Meetups and fairs played a pivotal role in advancing our human practices work by providing opportunities to gather valuable feedback, strengthen partnerships with other iGEM teams and external stakeholders, refining our approach, and further enhancing the impact of our project. Additionally, weekly tasks from the Biohackathon and Venture Creation Lab helped us explore the market potential of our project. Through this integration of expert insights, community outreach, and startup development, we ensured that our project is scientifically sound, social relevant, and impactful.
OncoBiotica marks a significant step towards a future where precise and effective therapies are the norm. By targeting the tumor's microenvironment, we stand apart from traditional therapies that focus on cell biomarkers. This innovation paves the way for highly personalized treatments that are more targeted and potentially less harmful. In combination with Mike, personalized cancer therapy can be transformed with an affordable and scalable solution for drug testing, especially for developing countries. With adaptability to various cancer types and the potential to advance treatment accuracy through data integration, Mike could definitely enhance cancer research and care globally, reducing healthcare disparities. With emerging research on tumor-associated microbes and the growing need for novel therapies, OncoBiotica is poised to revolutionize cancer therapy, bringing us closer to a future where each patient receives the care they need. Thank you for listening.
Thank you, guys. Thank you so much.